Yes, thank you.
Great. Okay, so thanks for inviting me. My name's Kiran, and this presentation is on development of bedside phenotyping approaches for acute hypoxemic respiratory failure and ARDS in general. So this is going to focus a lot on the FIND study, which we recently published, and then the follow-on work from that. So these are my disclosures. I guess the key one to point out is that FIND, which is the main study I'm going to talk about, was primarily funded by UK Research and Innovation, which is a government funder, but we did receive supplemental funding from industry. But this wasn't an industry study, so the funders had no input on design, conduct, or analysis of the study. So as a bit of background, acute respiratory distress syndrome, or ARDS, as we all know, is a disease of acute lung inflammation that's very heterogeneous. So there's a lot of different triggers for ARDS, and it's purely clinically defined by previously the Berlin definition and now by the global definition, which stratifies patients purely based on hypoxemia, chest imaging findings, and degree of mechanical support. And this tells us nothing about the underlying biology of the syndrome. And we probably are lumping together a bunch of heterogeneous patients by calling them all ARDS. So these are all of the pharmacotherapies that have been tried for ARDS in large phase 3 clinical trials. And the one thing that they all share is that none of them worked in these big trials after loads of good preclinical evidence and money was invested in these large scale trials. And they all resulted in net results of no treatment benefit. And one potential explanation for that could be this underlying heterogeneity of ARDS, which a lot of people are starting to pay more attention to in critical illness syndromes in general. So by treating all these patients with the same pharmacotherapy, we're probably lumping together responders. Whoop, did my slide drop?
Yeah, it just interrupted.
Can you see there again yet?
Yes, thank you.
Perfect. Okay. So probably by lumping together all these patients, we're bringing together responders and non-responders, and perhaps more worryingly, patients who are potentially harmed by therapies. So one approach we can take to try and deal with this heterogeneity is to better target subphenotypes of patients. So we can take a precision medicine approach to better identify potentially responsive subgroups of patients and split them into responders and non-responders. One of the problems with ARDS and acute hypoxemic respiratory failure in general is these subgroups are not readily apparent in the bedside in most cases. So this is a map of precision medicine approaches in ARDS that we wrote a while ago, and this is all the steps that we're planning on taking towards translating subphenotypes and precision medicine approaches to clinical practice. So it starts at the top with discovery of these subphenotypes, mostly in retrospective analyses and post-hoc analyses of completed trials, moving forward into the development of rapid assays or point-of-care tests that can identify these patients at the bedside, prospectively validating these assays and diagnostic approaches, and then using those assays to stratify patients into precision medicine trials that either use repurposed therapies or develop new therapies. So a bit of background, nomenclature, because it can be quite difficult in this field. There's a lot of kind of mixed up nomenclature. So what I mean by phenotypes in general, so phenotype is a group of patients that belong to a similar clinical syndrome. So in this case, phenotype could be ARDS or sepsis or hypoxemic respiratory failure. And then we split them into subphenotypes through clustering approaches or latent class analysis, which I'll get into, that identify patients with shared clinical characteristics and shared outcomes. And the idea then that we'd want to get to is get these patients towards, or would identify endotypes, which would be subgroups that share mechanistic patterns that we can target with pharmacotherapies. And then the ultimate goal beyond that would be to identify treatable traits, which would be syndrome agnostic mechanistic pathways that we can target. So if you could imagine an inflammatory pathway that we can target with the drug, and it doesn't really matter if you have ARDS or sepsis or acute hypoxemic respiratory failure, as long as you have that shared pathway, you can potentially all receive the same treatment. So this is all being preceded by seminal work Calfee in UCSF, who initially identified the hyper and hypoinflammatory subphenotypes of ARDS in 2014 now, so 12 years ago now, through retrospective analyses of the ARMA and alveoli trials, which were the big ARDSNet trials that identified benefit for lung protective ventilation and high PEEP strategies. So what Carolyn and colleagues did is they used biobank samples from ARMA and alveoli and measured a lot of different inflammatory biomarkers, which you can see on the bottom, and combined that with clinical data points and applied this approach called latent class analysis. So latent class analysis is essentially an unsupervised clustering approach, although it is not per se an unsupervised clustering approach, but it works very similarly. Structural equation modeling approach that assumes your distribution of data is made up of multiple hidden latent subgroups. And it's interestingly originally developed in the early 1900s by a biologist who used it to identify two species of crabs that were physically indistinguishable. So latent class analysis was applied to ARMA and alveoli. And Carolyn and colleagues put in all these ELISA inflammatory biomarkers as well as clinical data points. And what you find is you repeatedly get these two subphenotypes of ARDS, so the hyper and hypoinflammatory subphenotypes. So in the hyperinflammatory subphenotype comprises about 25 to 30% of the population, and it's distinguished by worse outcomes, so 50% mortality. Higher inflammatory biomarkers, more multi-organ failure, more metabolic acidosis, more shock, circulatory shock, more deranged coagulation. And then the hypoinflammatory subphenotype is the remaining 70-ish% of patients and has about a 20% mortality, 20% mortality rate, and is conversely defined by less circulatory shock, more single organ failure. So they tend to be more pneumonias or thoracic trauma, although it's not purely etiology-based distinction. And what you can see from this graph is these are the distinguishing biomarkers and clinical characteristics between the hyper and hypoinflammatory subphenotypes. So this is Z-scored standardized variables. So it's to make all the variables comparable on the y-axis. Things towards the left of the graph are up in the hyperinflammatory subphenotype, and towards the right, they're conversely down in the hyperinflammatory subphenotype. And then things in the middle don't really distinguish the two subphenotypes. So you'll note that the hyperinflammatory subphenotypes heavily driven by high blood interleukins, so IL-6, IL-8, soluble tumor necrosis factor receptor 1, plasminogen activator inhibitor 1, and then conversely on the right side, low bicarbonate, low protein C, low blood pressure, low platelets, so a lot of markers of multi-organ failure, shock, and circulatory and failure and deranged coagulation. So since this original study where the hyper and hypoinflammatory subphenotypes were identified, these have been repeatedly identified in thousands and thousands of patients across multiple trial cohorts, observational cohorts in ARDS, in sepsis, in patients at risk of ARDS, in all patients on mechanical ventilation. So these seem like they're very broadly applicable inflammatory subphenotypes that are highly replicable across broad categories of patients in multiple jurisdictions. And they were replicated again in this trial. So HARP2 was a trial in the UK of simvastatin in all-cause ARDS that was published in the New England in 2014 and found no benefit for simvastatin despite pretty good preclinical evidence suggesting that there would be benefit. And then if you go back and apply latent class analysis, you find that it looks like the hyperinflammatory subphenotype selectively benefited from simvastatin. So those two blue survival curves on the top are the hypoinflammatory subphenotype, placebo and simvastatin, and then the orange and red are simvastatin and placebo again. It looks like there was selective benefit in the hyperinflammatory subphenotype, which was probably diluted out in the overall trial because of the heterogeneity there. And I guess this is to bring up the other concept that I wanted to introduce that I had mentioned before with treatable traits, so that we'd hope that in future we can move away from a paradigm of defining critical illness syndromes using clinical symptomatology, so ARDS and sepsis and respiratory failure, and move more towards targetable biological pathways. And this whole process of identifying subphenotypes that are potentially distinguishable by inflammatory biomarkers, we would hope would lead us towards mechanistic insights that are targetable with medications and are not specific to syndrome. And here's some evidence to support that this could be possible. So this is from Pratik Sinha, who's also done latent class analysis. And in this case, he looked at Prowess shock, which was a sepsis trial of activated protein C that subsequently resulted in activated protein C being withdrawn from the market. And if you go back and you apply latent class analysis to this cohort, you find, again, the hyper and hypoinflammatory subphenotypes, and it looks like there was actually selective benefit for activated protein C in the hyperinflammatory subphenotypes. So you can see that there's reduced mortality as your probability of being hyperinflammatory increases. So we potentially threw the baby out with the bathwater in Prowess shock, and in hindsight, maybe better targeting of that medication of activated protein C based on inflammatory subphenotypes would have been beneficial. So to move on, past the background, so one of the big issues with latent class analysis, how we had currently defined or previously defined hyper and hypoinflammatory subphenotypes is that it's cumbersome. So latent class analysis requires knowledge of population means to be able to subphenotype these patients. And you need big data sets, you need lots of lab grade inflammatory cytokine measurements and knowledge of population mean, which means you essentially can only apply latent class analysis retrospectively at the end of a study or in post hoc analyses of completed studies, making it essentially useless for identifying patients at the bedside and potentially using that to stratify treatments. So how do we turn this into something that we can operationalize and use for prospective trials? So this is more work from Pratik, who developed a number of different parsimonious models, so simplified models for identifying the hyper and hypoinflammatory subphenotypes. So there's a number of different models here. Each row in this table is a model. And then across the top in the columns, are the variables that are included in each parsimonious model. So these are all logistic regression models that are trained to identify the hyperinflammatory phenotype. And in each one, the plus indicates that that's part of the model. So it's all some combination of 3 or 4 variables of either interleukin-8, bicarbonate off of a routine blood gas or a venous bicarbonate, soluble tumor necrosis factor receptor 1, vasopressor use, whether or not the patient's on vasopressors, protein C, or interleukin-6. And you'll see the one highlighted in red there is the one that we went ahead with in FIND, which is the study I'm going to show to you now. And that uses soluble tumor necrosis factor receptor 1, IL-6, and bicarbonate. And it has quite a good area under the receiver operating curve for identifying the hyperinflammatory subphenotype of 0.94. So you'll note there that some of these biomarkers are lab grade cytokines that are not routinely available in clinical practice, and then some of them are routinely available. So bicarbonate and vasopressor use are easy to get, and we measure that all the time in the ICU, whereas the other ones are a little bit more difficult. So the way that we operationalize this and FIND is we move forward towards using this 3 variable model, so the IL-6, STNFR1, and bicarbonate model, and we combined it with a point of care chemoluminescence assay, so the Randox multistat, which you see here on the left, which was supplied to us for the study by the company. And it's basically a benchtop chemoluminescence analyzer that essentially runs a robotic ELISA with the pre-coated plate and can multiplex both IL-6 and soluble tumor necrosis factor receptor 1 and give you a quantitative measurement of that from a blood sample within about an hour. So what we can do with this is we can use this parsimonious model to measure those two. So we can use the chemoluminescence analyzer to measure IL-6 and STNFR1 in an hour, combine that with bicarbonate off of a routine blood gas and get a patient's phenotype allocation within about an hour, and then potentially use that to stratify treatment in future. So this is just showing some data of the area under the receiver operating curve for the multistat, which is the red curve there. So you get an AUC of 0.89, which is still quite good. So we needed to then validate that we can actually do this prospectively in a real world setting and that this is something feasible. So this led us to take on the FIND study, which is the main bulk of this presentation. So the hypothesis of FIND is that we'd be able to prospectively define subphenotypes in patients with ARDS and with acute hypoxemic respiratory failure, so as an exploratory cohort beyond ARDS, using this novel point-of-care chemoluminescence assay and parsimonious model. And then we powered the study to 60-day mortality. So we hypothesized that we'd be able to identify the hyper and hypoinflammatory subphenotypes, and they'd have differing 60-day mortality in keeping with the retrospective data. So these are the inclusion-exclusion criteria. So it's a fairly sparse exclusion criteria. We wanted this to be quite a pragmatic, real-world study. I think one of the key things to point out as well, if you look at the inclusion criteria, so we defined ARDS by the Berlin definition as it was at the time, but we did actually also include patients on high-flow nasal oxygen. So this was much more in keeping with the new global definition of ARDS, which I think is a more contemporaneous definition of ARDS and reflects more real-world clinical insights at the moment. And we additionally included this group, AHRF, so acute hypoxemic respiratory failure, which we defined very similar to ARDS, except they had unilateral infiltrates on chest X-ray or CT versus ARDS that had bilateral infiltrates. So exclusion criteria excluded kids. We recruited patients within 48 hours of ARDS onset initially, and then extended that out to 72 hours partway through the trial based on pre-existing evidence for stability of the phenotypes. We excluded patients on ECMO. We excluded patients who were dying or had a DNAR order, declined or consent, or were incarcerated. So the primary outcome measure, as I said, was 60-day mortality in the hypo and hyperinflammatory subphenotypes in ARDS as prospectively defined by this point-of-care device. And we looked at another bunch of other secondary outcomes, so ventilator-free days, length of stay, duration of mechanical ventilation, which I'll show you. And we looked at agreement of the point-of-care test with gold standard lab phenotyping approaches using ELISA and using latent class analysis. We also looked at temporal stability between day 0 and day 3, and we measured feasibility of actually running this device in a real-world NHS and HSC setting in the UK and Ireland. So what we did is FIND ran in 30 centers in the UK and in Ireland, and patients with ARDS and AHRF were screened daily, and they were included if they gave consent, or we also had a deferred consent model in place for patients who had a next of kin that was unable to be contacted. Patients were phenotyped using the point-of-care assay on recruitment, and they also had biobank samples aliquoted and frozen and sent to a central biobanking facility in Queen's University Belfast, where we also did validation, cytokine measurements, and phenotyping at end of study. We also collected a number of DNA and RNA samples, as well as respiratory samples for mechanistic work. And then we followed these patients up to day 60 for mortality. So this is the strobe recruitment diagram for the ARDS cohort. So we screened just shy of 2,000 patients between 2019 and 2023. Most of those patients, you'll see 700 or so, were excluded for other reasons, and most of these reasons were actually COVID-19. So during the pandemic, there were a lot of sites that were unable to recruit COVID-19 patients because of staffing issues or because of biosample handling issues at the time when people weren't sure about transmission risk with COVID-19 samples. And then the next most common exclusion criteria, commonly met exclusion criterion, was timing after ARDS or AHRF onset. And then you have declined consent, do not resuscitate orders, treatment withdrawal, and ECMO. So we recruited 525 patients, which did meet our pre-specified sample size calculation. So our pre-specified sample size calculation required 480 patients to identify a 15% mortality difference between the two subphenotypes. And we inflated that number a bit to allow for some technical failure and loss to follow-up. So 525 patients were recruited. And what I want to bring your attention to is in the bottom right, the 22 that were excluded because they were unable to be phenotyped using the device. So this was quite small number of technical failures, which is reassuring and gave some useful evidence to show this would hopefully be feasible. So in the end, we ended up with 486 patients, including the analysis of the primary outcome, which did meet the pre-specified power calculation of 480. So the first result to highlight is that FIND showed us that real-time phenotyping is feasible. So these are the proportions of the hyper and hypoinflammatory subphenotypes as prospectively defined by the near patient assay and FIND. So we identified 490 patients with ARDS, and 401 of those were hypoinflammatory and 89 were hyperinflammatory, so just shy of 20%. And then these two subgroups you see below are ARDS patients on invasive mechanical ventilation specifically, which was a slightly higher proportion of hyperinflammatory patients, and ARDS patients on high flow nasal oxygen as defined by the new global definition, which had a slightly lower percentage of hyperinflammatory patients. So the baseline demographics of the ARDS cohort, I'll just leave them up there for a second so you can have a look. But overall, what this showed us was there's quite a bit of face validity lended here because the hyperinflammatory subphenotype had clinical characteristics that were in keeping with retrospective data from late in class analysis. So much more prevalence of sepsis, less pneumonia, more pancreatitis, less thoracic trauma. So much more kind of multi-system inflammatory processes rather than single organ lung injury. Much higher rate of renal replacement therapy use at recruitment. So 40% of these patients were on renal replacement therapy at recruitment. And lower incidence of COPD. So this was reassuring that we were identifying a very similar group of patients as was identified in late in class analysis and retrospective analyses. So this is the primary outcome here. So this is the money slide. We found that 60-day mortality was indeed different between the hyperinflammatory and hypoinflammatory subphenotypes as prospectively defined. So 50% mortality in the hyperinflammatory group, 27.9% mortality in the hypoinflammatory group. And this resulted in an odds ratio of 2.6 for worsened mortality in the hyperinflammatory subphenotype. And as you can see from the survival curves on the right, this mortality split happens quite early and is sustained with a continuous increased hazard ratio of worsened mortality for the hyperinflammatory subphenotype. Secondary outcomes were largely in keeping with the primary outcomes, so they were consistently worse in the hyperinflammatory group, so worse 28-day mortality, less ventilator-free days, longer length of stay in the ICU and the hospital in survivors. Interesting secondary finding here is that hyperinflammatory patients on high flow nasal oxygen were more likely to be subsequently intubated, so it seemed to be more of a predictor of high flow failure. Though caveat being there's small numbers here, so this requires prospective validation. We also did successful extubation analyses in the presence of competing risk of death. So hyperinflammatory patients were less likely to be successfully extubated with a hazard ratio of 0.54. And then the AHRF population, so this was the exploratory cohort of patients with unilateral infiltrates rather than bilateral infiltrates. We recruited less patients, so 131 patients, screening just shy of 500. And here are the outcomes from the AHRF group. So none of these reached statistical significance other than length of stay being longer in the hyperinflammatory subphenotype. So it looks from our data, at least, we can't definitively say that the AHRF phenotypes have different outcomes, whereas we can say that in the ARDS group. However, this might have been a result of underpowering, and this was indeed an exploratory cohort. We also did some retrospective validation work where we compared phenotyping by the prospective point of care assays, so the multistat, to lab assays, so Quantikine ELISAs and the ELA, which is an R&D also automated platform. And you can see on the left here, so these are the probabilities of allocation to phenotype. As you go up towards 1 on the x-axis, you're more likely to be hyperinflammatory, and as you go towards 0, you're more likely to be hypo. So on the x-axis, you have Quantikine probability, that's ELISA allocation retrospectively at end of study. And then on the y-axis, that's point of care validation prospectively. So blue dots are patients who were allocated to hypoinflammatory by both methods red dots are hyper by both methods, and gray dots are patients who are mismatched. So you can see there's a very good concordance between the quantikine and the multistat, with most patients who are mismatched falling around the decision threshold line of 0.5 probability for phenotyping there. Ella and quantikine were still quite well correlated, but less well correlated than quantikine and multistat. So this was some evidence for the use of other phenotyping approaches and other phenotyping platforms. So some limitations of FIND. You'll note that we found that there were 18.2% of patients with a hyperinflammatory subphenotype when we found them prospectively in FIND, which was less than what we had expected based on retrospective LCAs that had usually found around 25 to 30% of the patients were hyperinflammatory. So there's a few potential explanations for this. So one of them is that you'll recall that the high flow nasal oxygen patients with ARDS had a lower proportion of hyperinflammatory patients. So this could have potentially diluted the numbers down. We recruited patients on high flow in addition to patients meeting the Berlin definition, as opposed to these retrospective cohorts where exclusively patients with Berlin ARDS, so mostly on invasive mechanical ventilation, were recruited. The other thing is that this was a cohort study with quite pragmatic, permissive kind of inclusion-exclusion criteria. So there might have been a bias towards an overall less sick population than in a interventional trial where there would be an inherent bias to recruit sicker patients when you're giving experimental treatment. We also used a fairly permissive recruitment window. So we recruited patients up to 72 hours after ARDS or AHRF onset. And this was based on pre-existing evidence from latent transition analysis that demonstrated stability of hyper and hypoinflammatory subphenotypes over the first 72 hours of recruitment. But we now realize, probably in retrospect, that at least with the parsimonious model, timing is more important. So there is a good chance that some patients who we recruited that were phenotyped as hypoinflammatory may have been hyperinflammatory before they were recruited. So this resulted in adjustments to the trials that we'll be doing now, and I'll show you later. The intubation result on high flow, I think, is really interesting. So it supports a paradigm where we may be able to use hyper and hypoinflammatory subphenotypes to predict intubation failure, or predict high flow failure, excuse me, and maybe push for intubation earlier. But again, these are small numbers, and this needs to be replicated in a properly powered prospective study. So some discussions for future work and points for future work. So that one of the key questions here is, what is ground truth? Because there's not really a ground truth in this field as to, are quantikine ELISAs done at the end of study the truth, or is the multistat prospective allocation of the hyper and hypoinflammatory subphenotypes the truth? And does that actually really matter? As long as we're able to find patients with heterogeneity of treatment effect, and where is the optimal cutoff threshold for how we phenotype these patients, and should we shift that cutoff threshold depending on the medications that we're using? Is there scope for using additional devices to phenotype? So we've done some work on the R&D Ella, and I'll show you some other work in a bit where we're looking at other devices. When's the optimal time frame for identification? So I think after doing this study, I'm definitely more in favor of using a tighter threshold, because I do think these phenotypes are quite temporally dynamic. And is there scope for using other models? So I do think there's other ways that we can potentially use these phenotypes prospectively and target treatments. And there is a lot of work being done on developing clinical classifier models that use readily available clinical data points and potentially don't need to use biomarker or lab-grade assays. So the conclusions to take away from FIND is that we can now phenotype these patients. So FIND is the first prospective study to show that we can phenotype these patients using a point of care device, and it's feasible, and we can identify phenotypes with different clinical outcomes. And then the next step is to move on to a precision medicine trial where we actually try and use these phenotypes to stratify anti-inflammatory therapies, which is Panther, which is now open in the UK, and I'm going to talk to you about in a second. So meant to say, FIND was published in March, and just the other day this week, SPARK is the contemporaneous US study that did the same thing, and that was published as well in the Lancet Respiratory Medicine this week. So SPARK independently also identified the same things. So Spark used the Randox device as well. The difference being the Spark was a feasibility trial, so it was powered to feasibility rather than 60-day mortality. But they did find, again, that there was mortality difference between their hyper and hypoinflammatory subphenotypes. And I guess the interesting thing as well that I don't have up here on the slide is they also replicated that result of increased risk of high flow failure in the hyperinflammatory subphenotype. So on to the trials, so repurposing targeted therapies, which brings us to Panther. So Panther is a international phase 2 open label multi-arm adaptive platform trial that has started recruiting in the UK and will be starting in a number of other countries, hopefully quite soon. So Panther uses the hyper and hypoinflammatory subphenotypes as adjudicated at the point of care by the Randox multistat or by other devices, and allocates patients to anti-inflammatory medications based on phenotype. So the research question we wanted to ask in Panther was, do adults with ARDS with differing phenotypes respond differently to anti-inflammatory therapies? So the initial 2 medications that we're going to be using in Panther are simvastatin, so that's based on that retrospective evidence from HARP2 that I showed you earlier, and baricitinib, which is primarily based on evidence for benefit in COVID. Comparators usual care, and our outcome is 28-day organ support free days. Should be familiar from REMAP-CAP. So there's a lot of different outcome measures and endpoints. So just a few selected ones that I wanted to put up to show you what we're doing here. So we're looking at all subgroups of all this, the composite points of organ support free days, mortality at earlier endpoints, number of safety outcomes depending on the medication, length of stay. We're also looking at physical function at discharge, cognitive function, delirium, ICU acquired weakness, and long-term outcomes at 90 and 180 days. So this is an overview of the design of Panther. So it's quite busy, but just bear with me. If you look at the left, we're recruiting patients with ARDS, and we phenotype them into the hypo and hyperinflammatory subphenotype using a point of care device. They're then randomized in a 1-to-1 ratio to usual care or one of 2 active treatments. The first 2, which will be simvastatin and baricitinib, but there's a pipeline of treatments that are coming down the line, and there's an intervention prioritization committee that I'll talk to you about in a bit as well. So it's initially simvastatin and baricitinib, and then there's scope for declaring an arm efficacious or futile at predefined adaptive analyses. So the first adaptive analyses will happen at about 240 patients in each arm. And then based on pre-specified stopping criteria, medications could be deemed futile or efficacious, and then either shelved or graduated to a confirmatory phase 3 trial. So I'll remind you here that Panther is a phase 2 trial. The idea being the benefit would be mainly to rapidly screen multiple therapies and de-risk the ultimate phase 3. So you'll note the statistical triggers there. So we predefined an efficacy trigger of having a probability of an odds ratio of greater than 1.1 for improved organ support 3 days of more than 0.84. And then the futility threshold is probability of having an odds ratio less than 1.075 of greater than 0.78. So you'll note those thresholds are quite permissive. So there is and it's based on a phase 2 design. So the idea is to maximize efficiency. These triggers will not reach thresholds for regulatory approval, but they will allow drugs to be rapidly screened and move on to a phase 3. So these are simulation results for sample size calculation. So it's an adaptive trial that will have a variable sample size based on detected effect. We expect about 1,000 patients before we reach results in our first 2 treatments. The 80th percentile estimate for that is 1,200, or sorry, 1,200, and the projected maximum is just shy of 2,000 patients. And we've capped a maximum sample size for the first 2 therapies at 2,163 patients, and that capped maximum is based on the number of patients that it would theoretically take to get an answer in those 2 drugs, in those 2 subphenotypes in a standardized traditional randomized control trial. So Panther has a number of different committees. It's a big collaboration that I'm a small part of, and one of these arms is the Intervention Selection Committee. So Panther is meant to be really democratic and involve researchers from all over the globe. And one of the key mechanisms here is that intervention selection prioritization is as transparent as possible. So members of the trial team and academic partners and industry partners could all propose an intervention to be considered for entry into Panther, which is then passed to the intervention selection committee, who review it based on a number of predefined criteria for safety, pharmacokinetics, pre-existing evidence. They can then decide to pass that on to an independent review committee who will either agree with or disagree with their decision to prioritize the medication for the trial. And that can then be passed on to the trial steering committee and brought into the Panther pipeline. So Panther can have 2 active drugs at any given time, and we will then prioritize drugs that can be brought in afterwards. Panther also has built into it a large biobanking structure, so with multiple tiers of biobanking depending on the participating site. So one thing I think is really cool about this is there's so much we don't know about these phenotypes yet in terms of mechanism and potential treatment responsiveness, and the way we can develop ideal assays to better target these phenotypes and where we set the thresholds. So a big part of Panther is that it's going to be continuously generating biobanked samples that then can be used for mechanistic studies and validation work to potentially generate translational research that can then be fed back into the trial in the form of interventions. So there's 5 tiers of biobanking depending on the site's capability, with the top tier involving bronchoalveolar lavage and PBMC collection, tracheal aspirate collection, DNA and RNA collection, as well as biobanking plasma and serum. And then the tiers below depend on the site. And this will all be kind of managed in a bit of a distributed fashion, with regional expert hubs managing smaller sites and then sending on to a central processing facility. So as I said, Panther's a big team, and this is a picture from a Teams meeting a while ago of just some of the Panther team, and this is actually getting kind of out of date now. So this is a few years old now, and there's more people added, and people have come and gone. I think I want to point out there that the person I see there now that you'd miss is Tom Martin, who unfortunately passed away last year. And he's been a big part of Panther. He's actually the senior author on the paper from the figures that I was showing you before. Obviously, Danny and all the other leaders of the Panther trial, but it's a shame to see Tom had passed on. He was a big part of this, and he's a great guy. So how are we getting on with Panther? We're funded in the UK, we're funded in Canada, and in Canada we'll be part of the Practical platform. We're funded in the US, we're funded in Australia, and we're funded in Ireland. So all large grants for national leadership of Panther. Smaller pots of funding in Japan and Germany and France, and we're looking for funding in a number of other countries in South America, Italy, Belgium, and the Netherlands. And we have started recruitment in the UK in November of 2025. So we have sites open, and we're hoping to open sites in Canada and the US and Australia and Ireland before the end of this year. So this is from a scoping review that Ross McMullen in our group has done as well. So this is just to point out that Panther and the work we've done is not limited to one diagnostic assay. So this is a review of all the different devices that could potentially be used or repurposed for prospective phenotyping. So in FIND, we went ahead with the Randox device because of point of care ability and multiplex ability. But the way I see this, I think in future, if this works, we can hopefully start getting IL-6 and STNFR1 assays and other assays into routine clinical use on large scale, high throughput hospital analyzers. And we've certainly worked with companies like Beckman Coulter and Roche and Abbott to get this moved forward. And this just to show you the other interventions that have been considered for Panther and prioritized. So at the moment, we have simvastatin and baricitinib in the trial, and this is from our website. You can see online. I'll give you the link. We've also prioritized complement inhibition, atibuklimab, IL-6 inhibitors, TNF-alpha inhibitors, and then there's a number of other drugs under review that you can see there. So how do you get involved? Well, Panther, as I said before, is very keen on involving everyone who wants to be involved and involving and supporting early career researchers. So one of the subcommittees of Panther that I'm involved in is the Early Career Researcher Committee. And we have clearly defined early career research support strategies, recognition. So there's funds to support ECRs. ECRs all lead different subcommittees of Panther and are heavily involved in the writing, the work, the leadership, and taking the reins of Panther, because the idea being that this is hopefully going to be a perpetually running platform trial, and it's going to be passed on to the younger people at some point. So thanks very much for listening. There's a couple of QR codes up there. So FIND is the study that I was talking about, and that was published in the Lancet Respiratory Medicine in March. And then Panther, if you look at that QR code, that'll bring you to the Panther website and show you all the different therapies that we're looking at prioritizing and ways you can get involved in the ECR committee and other aspects of Panther. Thanks very much.
Thanks so much, Kiran. I think we have time for some questions. Feel free to write in the chat or raise your hand. And while we're allowing people a chance to put their questions down, Kiran, maybe I'll just start. So really exciting work. Very exciting to think that we're not too far from having biologically driven prospective clinical trials, and showing the sort of information on the devices is quite interesting. It sounds like you're pretty optimistic in terms of these devices being ready for prime time, maybe not in terms of actually launching trials, but at least to be picked up by other institutions and be involved in some of this work, at least from a feasibility standpoint. Is there, in your experience, are there sort of standardized SOPs or anything in place that would be disseminated to other institutions that would be interested in doing this? So, for example, if I have a lab, I have a centrifuge, I have some personnel that can help sort of collect samples and spin this down. Can I just get started, or is there sort of, information package or SOP, something that can be disseminated to make sure that these results are standard and up to par to expert sites?
Cool. Yeah. So in FIND, in the study that we did there for prospective validation, we did this in a very kind of rough and ready sort of way. So at site, it would have been research nurses collecting blood and spinning it down and running it on the device. And these would have been mostly research nurses without much prior lab experience. So we really wanted to make this as pragmatic and demonstrate feasibility as kind of a point of care assay in the study. So as part of FIND, we did distribute SOPs, and then Panther, we do have sample handling SOPs, and that's all available on the website. So for the Randox device itself, it's pretty simple. There's just a plate with like a tinfoil top, and you just kind of poke a hole in it with the pipette, and then you put in 400 microliters of spun plasma into the plate, put it in, and press go. So I guess there's basic centrifuge management, but it's not that hard to do. But I suppose, yeah, you get into other questions as well. So we've looked at doing the ELA. So we use that prospectively, and we compared it to multistat and to quantikine, and it looks like it works pretty well as well. I know you and I were talking beforehand about the ELA, and it's also a very good device, very easy to use. Just one simple kind of centrifuge step. I guess the thing with the ELA is that it requires one dilution step as well, which could be a stumbling block, but it shouldn't be too bad. So they're relatively easy to use. They can be done in a clinical setting with people without much previous lab experience. But I suppose going forward, this kind of brings up the question of other assays and how you compare all the assays to each other, and what do we define as being gold standard, and what's the best way to do it. And I think you can get really into the weeds here, because for these assays, for IL-6 and STNFR1, there is no industry gold standard assay. So the way you get into it is comparing phenotype allocations and trying to decide, which ones are the most concordant and how they concord with your new assay that you want to validate. And I guess for me, what matters the most to me is that we can identify patients with heterogeneity of treatment effect, I think is the bottom line, right? When you're in this kind of a field where there's no clear gold standard. Probably what really matters is you're identifying patients that you can actually make a difference to with therapies. But I think, yeah, ultimately there will hopefully be more assays brought in, and I can see this hopefully being moved more towards, high throughput automated analyzers in the hospital lab. So there are IL-6 cartridges available for, the Beckman Coulter devices and for the Abbott and the Roche and the Fuji Raybos. But there is no